Welcome. I'm Kathy Engelmeyer. I'm the Director of Principal Gifts at the Wilmer Eye Institute. Thank you for joining us as we honor and celebrate the remarkable legacy of Lois Zenkel, who I'm sure is with us in spirit today. Her philanthropic partnership with Dr. Thomas Johnson has been instrumental in advancing his work and bringing us all together in the quest to curing blinding eye diseases. Today's webinar will conclude at 5 p.m. You will have the opportunity to post questions in the chat. Now I would like to invite another dear friend of Wilmer's, Alan Holt, to offer a brief introduction of Dr. Johnson. Alan?
Thank you, Kathy. Good afternoon. Thank you for joining us for this Lois Zenkel Memorial webinar in honor of Lois. I'm Alan Holt. I'm a proud member of the Wilmer Advisory Board, and along with my wife, Shelley, we are honored to introduce Dr. Thomas Johnson III, the Shelley and Alan Holt Rising Professor of Ophthalmology. Dr. Johnson is both our presenter and a beneficiary of Lois's kindness and generosity. Dr. Johnson is a clinician, scientist, glaucoma specialist, and translational neuroscientist at the Wilmer Eye Institute. He grew up in the suburbs of Chicago, completed his undergraduate degree at Northwestern University, studying chemistry and biology, and attended Johns Hopkins School of Medicine for his medical degree. He also completed his PhD, funded by the Gates Cambridge Scholarship at the University of Cambridge in the UK. After earning his PhD, Dr. Johnson began his clinical training at Johns Hopkins, where he encountered an unparalleled environment of mentorship and support for trainees. Throughout this time as a medical student, an intern on the Osler Medicine Service, a resident at the Wilmer Eye Institute, a fellow in the Wilmer's Glaucoma Division, and a year serving as the Wilmer Assistant Chief of Service, Dr. Johnson was nurtured by an incredibly generous group of Hopkins and Wilmer faculty. Now a fully trained physician, scientist, and emerging leader in the field, Dr. Johnson runs a translational neuroscience laboratory aimed at developing innovative treatments capable of not simply preserving vision, but actually restoring sight and reversing blindness in an array of optic nerve diseases. Dr. Johnson lives in Maryland with his wife, Stacy, and their two children. And it is our honor to introduce him for this one webinar. Tom?
Well, thank you very much, Alan. I really appreciate it. I'm going to share my screen so that everyone can see some slides and some material that I'll be using to discuss today. So the title of this lecture is Prospects for Restoring Vision in Patients Suffering from Optic Neuropathy. And as you know, this is the Lois Zenkel Memorial Lecture. Now, I want to do three things this afternoon in discussing with all of you. First, I want to honor the memory of a very special person, Lois Zenkel, who we sadly lost this year. But in addition to that, I also want to help educate everyone that's on the call. We have a diverse array of people that have joined us here today, some of which are friends and family of Lois Zenkel, some of which are patients of mine and of other doctors here at the Wilmer Eye Institute, and their families and friends and caregivers. And I want to describe optic nerve disease and some of the specific diseases like glaucoma and ischemic optic neuropathy in a way that I think doctors and their patients don't always think about these things in the clinic. In the clinic, we tend to focus on big picture items. In glaucoma, for instance, we talk about what's my eye pressure today? What does the visual field look like? And maybe what does the optic nerve look like? But we don't always get into the real nitty gritty details that help patients understand what the optic nerve really is and why that's so important to vision. So we're going to learn a little bit about that today. And then finally, I do want to update everyone on the research that we've been doing with the goal of not just being able to preserve vision for patients suffering from optic neuropathy, but actually to restore vision in this class of blinding diseases. So I want to begin with a tribute to Ms. Zenkel. I had the pleasure of knowing her for the last couple of years. She was an amazing person. She dedicated her entire life really to serving others. She was an accomplished photographer and art collector in her own right, but she spent much of her life engaged in civic organizations and in politics, and she served on many boards, as you can see here, and philanthropically supported a number of worthy causes in both science through our work, but also education. She spent her life trying to make the world a better place, and I think it's a real tribute. The work that we're trying to do, I think, in her memory, is also trying to make the world a better place. So I want to thank the Zenkel family for being here and for going on this journey with us. So now we'll get into a little bit of the educational aspect of this lecture. I want to begin by answering the question for everyone: what is optic neuropathy and why does it cause vision loss? Well, first of all, optic neuropathy is a very general term that just means it's a disease that causes damage to the optic nerves. There are actually many different types of optic neuropathies. The most common all over the world is glaucoma, and that's the one that I treat as a glaucoma specialist. Ischemic optic neuropathy is another type of optic nerve disease that is also very common and was near and dear to Lois Zenkel. And that is a different disease that causes nerve damage through blockage of blood vessels that supply the nerve. Anecdotally, it's sort of like having a stroke in the optic nerve rather than in the brain. There's also inflammatory causes of optic nerve degeneration, compression by tumors. There's a number of systemic diseases, autoimmune disease, infections, and cancer that can infiltrate the optic nerve and cause damage to that structure. Patients with severe head trauma can develop a traumatic optic neuropathy. There's inherited and genetic optic neuropathies. Patients may just be unfortunate to be born with a genetic disease that leads to blindness over the course of their lives. And then there are a number of toxic materials and nutrient deprivations that can cause optic nerve disease. To go into a little bit more detail about the two most common optic neuropathies, glaucoma is a disease where the optic nerve is damaged very specifically by high eye pressure. So the eyeball is sort of like a water balloon, and it's constantly creating internal fluid to keep itself inflated. And then it's draining that fluid out of the eyeball and into the blood vessels that surround the eye. And this is a very delicate balance of inflow and outflow, kind of like a bathtub that is constantly flowing, but also draining to keep the water level balanced. If the outflow pathway becomes blocked, then the pressure can build up inside the eyeball. And that pushes on all of the structures inside the eyeball, but the optic nerve, the location where the nerve enters the back of the eye, is the most susceptible to this pressure. And so the optic nerve is the first thing to become damaged as pressure builds up inside the eye. Now, separately, as I alluded to, ischemic optic neuropathy is when there's a lack of blood flow to the optic nerve as it enters the back of the eye. We'll get into in a second what the optic nerve is, but it has a very rich supply of blood vessels that deliver oxygen and sugar and other nutrients to the cells in this location. And if the blood flow gets cut off, then this tissue becomes what's called ischemic, meaning it is deprived of oxygen. And again, it's sort of like having a stroke in the optic nerve in the back of the eye. Now, when the doctor looks in the back of your eye with that super bright light at the slit lamp, one of the things they're looking at is the optic nerve itself. It's directly visible in the back of the eye at the location where the nerve enters the back of the eyeball. And this is what a healthy optic nerve is supposed to look like. It's sort of an orange disc. What you're looking at is a cylinder, but you're looking at it side on. So you only see the circle in cross section. And out of this orange disc comes blood vessels. This brownish tissue around the nerve is the retina. We'll talk about that in a second. But it's supposed to have a nice orange color, a thick rim of tissue here. This is nerve tissue, sort of like brain tissue, that is communicating vision from the eye to the brain. Now, when patients have glaucoma or ischemic optic neuropathy or other eye diseases, the appearance of this nerve changes, and that's what your doctor is looking at. When a patient has ischemic optic neuropathy, at the very time that that blood vessel blockage occurs, you see swelling of the optic nerve head and blood that can occur at the optic nerve and within the retina. As this resolves with time and the optic nerve degenerates, it goes back to looking a lot more like a healthy nerve, but it becomes much more pale. It has a whitish or light yellowish color, indicating that the nerve fibers in this nerve have died. Glaucoma is a little bit different in that the nerve doesn't change color, but the rim of healthy tissue disappears. And in 3 dimensions, this nerve actually adopts a cupped appearance. So for any glaucoma patients or their families, you may hear the doctor talking about the cup to disc ratio or the amount of cupping that a nerve has. And this is how we judge, in one way, the severity of someone's glaucoma. Now, there's a couple of key takeaways about all optic neuropathies that explain why this is such a devastating class of diseases. First of all, for all optic neuropathies at this point in time, we have some ways of trying to slow down or halt the disease from getting worse. And we can do that in glaucoma by lowering the eye pressure, in other diseases by reducing the risk factors that patients may have had to cause the disease in the first place. But the treatments are not 100% effective in all patients, and some continue to lose vision despite our best efforts as doctors. And many patients are diagnosed only after severe damage has already occurred. In glaucoma, this occurs because the vision loss is very insidious. It's very slow. It affects the peripheral vision first. And so patients don't notice it right away. In diseases like ischemic optic neuropathy, the blockage of a blood vessel comes out of nowhere and happens instantaneously. And this is essentially never caught until it's already happened. And by that time, in some ways, the horse is already out of the barn. Now, let's talk a little bit about these structures I've been discussing, starting with the optic nerve. So this is a cross-section of a patient's head. These are the bones of the eye socket. And so you're looking at the head from side on. This is the eyeball with the cornea in front. And this yellow structure coming out of the back of the eye is the optic nerve. The optic nerve is the main conduit that connects the eyeball to the brain. So this yellow structure goes through the back of the eye socket and into the skull. And what you're looking at on the right is a diagram that demonstrates how the nerve fibers of the eyeballs actually enter the brain and then communicate with very important, very specific centers inside the brain that are important for our vision. Now, the optic nerve is not really a single structure. It's actually a cable that is made up of hundreds of thousands or sometimes millions of little fibers called axons. So there's going to be a little bit of scientific nomenclature in here, but I'm going to try to explain this as clearly as I can. I think it's important, though, to really understand on a detailed level what we're trying to do in the lab. So what you're looking at here on the left is a retina of a mouse, and it's a genetically engineered mouse that's been made to label some of its retinal ganglion cells in blue. Retinal ganglion cell is abbreviated RGC. And you're looking here on the right at just a magnified view of a handful of these RGCs within the retina. Now, the RGC is a type of neuron or nerve cell that I'm going to be talking about a lot because it's central to what's happening in patients with optic nerve disease. As you can see from this picture here, a retinal ganglion cell is really kind of a beautiful and also complicated cell type. It has a little dot in the center, and it has all of these branched finger-like projections coming off of the end of it. Some people think these looks like the branches of a tree, and so this is actually sometimes referred to as a dendritic arbor. The other thing you'll notice is that coming from each of these cells is a single long fiber that goes from wherever that cell is located in the retina towards the very center. And this right here is the optic nerve head. So the optic nerve is composed of all of the fibers or axons of these retinal ganglion cells as they exit the eye. Now, a retinal ganglion cell plays a critical role in how we perceive the world around us through vision. So on the left here, you have two pictures. There's a schematic diagram of the retina, and then there's a histological or microscopy slide of a piece of retinal tissue to show you what this really looks like in a tissue. I want to explain how vision, how light entering the eye gets turned into a picture that we perceive in our brains. The first thing that happens is that little pockets of light energy called photons enter the eye, and they're shown on the back wall of the eye. In some ways, people like to compare the eyeball to a camera. It's got lenses in it that focus the light, and then the back wall of the eye is sort of like the film on a camera. When the light hits it, it turns that light into a picture. Now, the retina itself is actually composed of several different types of nerve cells. The ones that absorb that light energy and turn it into an electrical signal are called photoreceptors. And you've probably heard of these more commonly referred to as rods and cones. So the rods and cones absorb light energy, they turn it into an electrical signal. And then they pass that on to a middle type of cell shown in pink here called bipolar cells that process that light information a little bit. And then they pass it on to the cells that we're talking about today, the RGCs or the retinal ganglion cells. So the RGCs receive that information about light entering the eye, and then they pass that information along their axons through the optic nerve and into the brain. So optic neuropathies cause vision loss specifically because they kill RGCs or retinal ganglion cells. The other cells in the brain and in the retina are pretty much unaffected by most optic neuropathies. It's only the retinal ganglion cells that die. And when that happens, this disconnects the eye from the brain. So the goals of our work are to restore vision for patients suffering from optic neuropathy. And if we're going to do that, we need to replace retinal ganglion cells in the eyes and optic nerves and brains of patients that are suffering from these diseases. This is a schematic of a review article that we wrote in the lab about 3 years ago and shows the major milestones of what we will need to achieve in order to restore vision for patients with optic neuropathy. We need to be able to create retinal ganglion cells, and we can do that now from stem cells, as I'll talk about in a little bit. We need to be able to get these from a petri dish into patients' eyeballs. And then once they're there, they need to survive. They need to remain healthy. They need to integrate into the patient's retina and then form communication stations between the retina and then all the way in the patient's brain so that that conduit of information can be restored and vision can be returned. Now, I want to go into a little bit more detail about exactly what it is that retinal ganglion cells are doing in the optic nerve in order to connect the eye to the brain and provide a visual picture of patients about the world around them. So I'm going to go back to this picture here where we're looking at a number of retinal ganglion cells in a mouse's retina. And on the left is a stylistic rendering of one of those retinal ganglion cells. So again, the headquarters for this cell, where most of the action is done in the DNA housing and the creation of proteins and energy, takes place in the cell body. But the communication that retinal ganglion cells perform has much more to do with the processes that come off of the cell body. So these finger-like branched projections that surround the cell body within the retina, these are called dendrites. And dendrites are essentially the listening end of the retinal ganglion cell neuron. This long fiber, and there's usually only one, that comes off of the cell body and travels very long distances to get to the brain, are the talking end of the retinal ganglion cell. So I want to give you a couple of analogies to help understand how this works. The first one is that childhood game of telephone, where one child in a line would whisper a word to the second child, and then they would have to hopefully hear the correct word and then whisper it on to the second child. And then they would whisper that word and so on and so on. And usually because of the children mishearing each other, by the time it gets to the end of the line, there's been some confusion or change in what that word was that whispered. So this would be an example of passing along a signal, in this case a word, but in the case of the optic nerve, signals about vision, in a way that eventually the last person in line, which is the brain, would need to understand. Now, here's a little bit of a more sophisticated analogy, and that is one of the TV in your living room. Now, this could be a little bit complicated with a lot of wires and boxes, and a lot of things can go wrong with this. But let me take you through this. When you have a TV, usually in this day and age, you won't get a picture on the TV unless it's plugged into a cable box. And that cable box needs to plug into your wall, and then it needs to connect to the TV. Now, the cable signal comes through the outlet, it goes into the cable box, and then it's connected by a wire, and that's what gives you your signal on the TV. This is, in some ways, exactly how the eye and the brain work to form a picture in your brain about the world around you. The TV is essentially like your brain. The rods and the cones are like the cable outlet. This is what's taking the raw material that eventually forms the signal that's going to come into your brain or your TV. Your retinal ganglion cells are like the cable box. They take this raw information coming from the cable outlet and turn it into a signal that the TV can understand. And most importantly, your optic nerve is the cable that connects your cable box to the TV. This is the listening side of the retinal ganglion cells. They need to have a wire or some kind of connection that plugs them into the cable outlet in order to adequately receive communication about vision. And this is the talking end of the retinal ganglion cells, where they need to send a signal out to the TV so that you can get a picture. And just to let this analogy hit home, if we reverse this, you have your brain here, which is the television. You have the rods and cones in the back of the retina, which is the cable outlet in the wall. You have the retinal ganglion cells up here, which is your cable box. And then the optic nerve connecting the eyeballs to the brain is your HDMI cable. So what is that optic nerve really? We talk about it as though it's one structure, but it's actually a very delicate and complicated structure. Now, I hope this isn't too graphic for people on the call, but this is a dissection like medical students might perform in a cadaver lab. where the person taking this photo has isolated the optic nerve and then cut it in cross section so you can look into the optic nerve and see what's actually here. And you'll see that there's a sheath of kind of fibrous hard material on the outside that protects the nerve. There's a spider web layer of tissue underneath that. And then this yellow blob in the middle is the actual optic nerve itself, the parenchyma of the nerve. And while this looks like just one solid structure, if you were to look at this under the microscope, it would look something like the images on the right side, that in high magnification show you there are actually hundreds of thousands of little circles within this yellow stuff in the middle of the nerve. And every single one of these tiny little circles is a retinal ganglion cell axon. So in some ways, the optic nerve, the analogy I just gave you for it being an HDMI cable is a very apt one, because the optic nerve is sort of like a fiber optic cable. If you cut a fiber optic cable, it's actually got a whole bunch of tiny little filaments inside it that each communicate a unique aspect of the signal that's trying to go through the wire. And the optic nerve is sort of like that, where each of the filaments is a retinal ganglion cell axon. Now, how do RGCs actually receive and send communication to other nerve cells? So there's one other scientific structure we need to talk about, and that's called a synapse. A synapse is essentially a communication station between two nerve cells. So I have a picture here of two nerve cells, a green one and a blue one. The green one is sending a message to the blue one. And in order to do that, it actually has an area where the tips of the axon, the talking arm of the green one, are very, 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 very close to the tips of the dendrites, or the listening arm, of the blue cell. And if you looked at this in high magnification, this is a stylistic rendering, but it would kind of look like this. These are the two tips of these neurons coming together. There's a tiny little space between those tips called the synaptic cleft. And in the middle of these, you see a bunch of little balls. These balls are vesicles that were produced by the green cell. And when the green cell wants to send a message to the blue cell, it puts these little balls out into the synaptic cleft, where they travel and then get taken up by the blue cell. These little balls, these vesicles, have substances in them called neurotransmitters. And that is what essentially mediates or carries the signal between the green neuron and the blue neuron. So RGCs, in order to perform their duties as communicators of visual information from the eye to the brain, have two sets of synapses. They have input synapses, where they receive information about light, and those synapses are located in the brain between the retinal ganglion cells and the bipolar cells, in a layer of the retina that's called the inner plexiform layer, or the IPL. This is the listening arm of the RGCs. The output synapses are located very, very far away from that. They're actually outside the eyeball and in the middle of the brain. And here, the RGC's axons are sending information to a part of the brain called the lateral geniculate nucleus, or LGN. But this is how the information about vision from the eye initially gets into the brain so that you can see. Now, what exactly is happening in neurons that allow them to understand communication and to communicate to other cells? Well, nerve cells have this really special property in that they are essentially electrically active. They can conduct electricity. And this is one way that we represent the electrical state of a nerve cell. We measure the voltage of the cell over time. And when a cell is trying to communicate information, the voltage in that cell will change very quickly. It's not important to understand the details of this. You can kind of think of this as though the nerve cell is experiencing a very short, very tiny electrical shock. And that is a form of communication for nerve cells. Now, if you take this one tiny little electrical shock and look at what a nerve cell is doing over much longer periods of time, each of these little shocks, which we call action potentials, looks like a little line or a blip. And if you follow the communication that a nerve cell is giving over a period of maybe 10 seconds, like it's shown here, you see a bunch of blips in a row, and then a period with no blips of silence, and then some more blips, and then nothing, and then blips, and then nothing. And different neurons will have different patterns of action potentials or blips, depending on what it's trying to communicate. So at the end of the day, the way that neurons communicate information is sort of like Morse code. They are firing action potentials. These little electrical shocks come in pulses that vary in how fast they are and how rapidly they follow each other. And this encodes something. But instead of dots and dashes encoding numbers and letters, the blips of neurons, of retinal ganglion cells, for instance, encode the picture that the eyeball is trying to focus on. So now, with all of that as the background, I want to tell you about some of the work that we've been doing and that has been made possible by the Zenkel family over the last several years in trying to replace retinal ganglion cells to restore vision for patients suffering from optic neuropathy. And I'm going to start with the punchline here and then take you through how we've made these conclusions. The punchline, though, is something I'm pretty excited about. We have successfully transplanted human stem cell derived RGCs, or retinal ganglion cells, into eyes and gotten them to form new synapses between the donor neurons and the recipient retina. So not only have we gotten these neurons to survive in living eyeballs, but they're actually engaging in the process of communication that is taking place and what needs to take place in order to actually restore vision. So I'm going to show you a video here. This is from an experiment in my lab where we took human stem cells and we turned them into RGCs, or retinal ganglion cell neurons, and we forced them to express a red fluorescent protein so that we could see them easily under the microscope. We transplanted those into the eyes of mice and then looked at the tissue. And so what you're looking at here is actually a small block of retina. And because the retina is some thickness to it, you can't just look at this on microscopy slides. You have to look at the tissue as a three-dimensional structure. And when we do that, you'll notice that these human RGCs survive very nicely. They even extend processes that might be dendrites or axons, but they're sitting on the surface of the retina. They're really not sending their processes down into that layer that they have to in order to form synapses with the other cells and receive information about light entering the eye. This was the very first attempt that we made at transplanting retinal ganglion cells. And while we were excited that the cells survived, we recognized that there was clearly a challenge here, because if this was all that we could achieve with retinal ganglion cell transplantation, we wouldn't be able to restore communication between the eye and the brain. So we did some work and tested a hypothesis that there's actually an anatomical structure on the surface of everyone's retina that serves as a fence or a barrier, so that when we inject human stem cell derived neurons into the eyes of mice, this fence prevents them from interacting with the retina. We developed several methods in the lab to poke holes in that fence. We found some enzymes that could chew little holes in the membrane itself. And we also identified a type of transgenic mutant mouse that has a developmental abnormality, that it develops a membrane on the surface of the retina that naturally has holes. So for proof of principle, we tested the hypothesis that if there were holes in this membrane, maybe that would enable our transplanted neurons to get into the retina much better. And this is the result. This is one of my favorite pictures still to come out of my lab. This is a low magnification image of a mouse's retina after we've transplanted human red neurons into the eye and disrupted the membrane that was acting like a barrier to their engraftment. Now, in this image, you can see that these human cells survive for several weeks inside the mouse's eye. They extend little dendrites around the cell body. And what was most amazing is that many of them extend a long process, an axon, that somehow knows exactly where in the retina it needs to go in order to get to the optic nerve head. This is the optic nerve head right here. And if this were 3 dimensions, the optic nerve itself would be running out the back of your computer monitor right now. Now, of course, we wanted to look at this in higher detail, so we did some more of those fancy 3-dimensional reconstructions of the tissue. And unlike the case where that membrane was intact and the cells could not integrate into the retina, if we permeabilized this barrier on top of the retina, the transplanted neurons could actually get in. So here's an example where there's 4 retinoganglion cells, and one of them, this one right here, managed to crawl through a hole in the membrane and get into the retina. So we've traced it in gray so that you could see it easily. And now if you look at this cell, its dendrite processes, the listening ends of this neuron, have gone down into the retina, and they're in this layer right where those input synapses between RGCs and the other cells of the retina have to occur. So we've managed to get retinoganglion cells into the correct part of the retina so that they could actually receive information about light entering the eye. Now, the next question was, are they actually making connections with the other neurons of the retina so that they can properly receive information? Now, we had several questions about this. First of all, when we looked at the profile of the cells we transplanted into the eye, we noted that they had many of these little processes that branched to some extent, similar to the dendritic arbors of normal RGCs I showed you earlier. But we had a question of whether the cell knew that any of these was supposed to become a dendrite, a listening process, or an axon, a talking process. We were able to use some clever tools in the science lab that helped us answer this question. And part of this relies on the fact that different parts of cells will express genes or make proteins that are unique to the area in the cell in which they're found. Dendrites, for instance, will make a special protein called MAP2 that's found in the dendrites but is not found in the axon. Similarly, axons will make a special protein called SMI312 that is found in axons but is not found in dendrites. And so by using antibodies to label the location of these 2 proteins in our transplants, we were able to tell if the cells that we had injected into these mouse eyes had decided whether certain processes were destined to become dendrites or axons. And this is a result of some of that work. So this is a technique where you label a protein by using an antibody. The antibody turns that protein a fluorescent color. And you can do this with multiple different proteins on the same sample. And so up here in pink is a label just showing all of our retinoganglion cells that we transplanted into the eye. And in yellow is a label for the dendritic marker MAP2. And in blue is a label for the axonal marker SMI312. And it turns out that all of the tiny little processes right next to the cell body that had gone into the retina in the correct location to form synapses, expressed MAP2, the dendritic marker, but not SMI312. And the opposite was true of those very long processes that coursed towards the optic nerve. Those showed signs of being axons, but not dendrites. And so our transplanted cells were correctly maturing and specifying the parts of the cell that would be in charge of receiving information or sending it on to the brain. Now, we next asked whether our donor cells are forming synapses or communication stations within the retina. We did this first by, again, using that antibody technique to look for little tiny components of the machinery that has to be present at these synapses. These are proteins that are involved in either putting out those spheres, those vesicles that contain neurotransmitters, or the receptors that allow neurons to receive those vesicles and use that to turn that into an electrical signal. And we were able to identify many little communication stations or synapses that were present on the dendrites of these donor retinoganglion cells within the retina. But what I'm most excited about is some data that we've just managed to obtain over this last summer. And this was based on our asking of the question, are the donor RGCs actually communicating? Are they performing a function that shows they are receiving information about pictures that are being shown to the eye? Now, answering that question is scientifically a really difficult thing to do. The traditional way of knowing whether a nerve cell is communicating is to do something called whole cell electrophysiology or patch clamping. And to do this, you grow neurons in a petri dish, and then under a microscope, you use a super tiny needle to impale the cell. And that needle is connected to an electrode that can measure the voltage in the cell so that you can actually record those tiny little electrical shocks that are going on in a cell. The problem is, this is very tricky to do. It's very time consuming. You might spend an entire day getting data from one or two cells. And if we're going to replace retinoganglion cells in human patients, our goal is to do this in thousands of cells. So we turned to a more cutting edge technique called optical electrophysiology. Now, this is a very cool method that allows us to understand the electrical impulses within nerve cells simply by looking at them. And it's based on a class of genes and proteins called fluorescent proteins. You may be wondering why I'm showing you a picture of green jellyfish. And the answer is interesting. Many years ago, there were some very clever scientists that noted some jellyfish in the ocean glow certain colors when special light is applied to them. They studied this phenomenon and determined it's because these jellyfish have a special gene in their chromosomes that create a protein called green fluorescent protein. And an intrinsic property of that protein is that it glows green. Over the subsequent years, the scientists were able to genetically engineer green fluorescent protein to turn it all different colors of the rainbow. And this has become a mainstay of what we do in the lab. Fluorescent proteins unlock a number of really powerful tools for understanding biology and the way cells work. There are now genetically engineered fluorescent mice and fish and flies and other animals. And since it's, if you're following the news, it's Nobel Prize season right now. This discovery was actually awarded the Nobel Prize in 2008. So we've been leveraging this discovery and a late-breaking engineering principle based on this to visualize the electrical activity of our transplanted cells. The idea here is that there's a special gene that's a derivative of green fluorescent protein. It's called GCAMP, but the name is not that important. The idea is that it is fluorescent, but it's not always fluorescent. The amount of fluorescence, the brightness of the signal that you get from this molecule under the microscope is directly related to how electrically excited the neuron is that contains that marker. And so when we force our retinoganglion cells to express this fluorescent protein, we can take microscopy images of it and look at all of these cells over time. But if instead of just taking a snapshot, we actually take a movie of these cells over time, we can see the fluorescent brightness of these cells change in time. And when those cells become brighter, we know that they are electrically active. They're firing action potentials. And when they're dim, we know that they're quiet. They're not firing action potentials. And that is shown here on this graph. For each of these lines, this is a different cell in this picture to the left. And over time, right at the 10-second mark, you could see that a number of them become much brighter and then go dim again. And that's because at the 10-second mark, we flashed a picture at the retina that these cells were living inside. And what you're seeing here is actually a description of the Morse code used by the retinoganglion cells to interpret the picture we just showed the retina and communicate that onto the brain. Now, if a picture is worth 1,000 words, I think a movie is worth 10,000 words. So this is one of those time-lapse movies of a bunch of retinoganglion cells here. And you can see that for most of the movie, they're kind of dim. And then every 10 seconds, they get very bright. And again, this is the fluorescence of that optical electrophysiology marker. And so every 10 seconds, we're seeing the electrical activity of these retinoganglion cells as they receive information about a picture entering the eye. So it took us a better part of a year to develop this technology, to use it in the lab, and to be able to understand how retinoganglion cells in the eyes of mice and other models are interpreting pictures. But we now can take images of a mouse's own retinoganglion cells and show pictures to it and see the Morse code output of those cells every time we show it a picture, knowing that these retinoganglion cells are receiving the information about that light from the rods and cones in the back of the eye. And what we're so excited about is, having established the technique, we then transplanted human stem cell derived retinoganglion cells into the eyes of mice and did the same experiment. And these human RGCs actually responded to the picture that we showed the retina. So this proved to us that the cells were not just structurally in the correct part of the retina, but they were actually functional. They were receiving communication about the light entering the eye. Now, we are what I call a translational neuroscience laboratory. Some science laboratories primarily do their experiments in order to better understand the world around them. And while that is important to us, our primary motivation, our drive, is to improve the lives of patients. So we're always thinking about how can we take the knowledge that we've gained in our lab and turn it into new treatments that will increase or improve the quality of life for our patients at the Wilmer Eye Institute. So we've done a couple of other experiments that I think are really important on the path to clinical translation, to bringing this into the clinic. One thing is, what I've been telling you about so far has been experiments in laboratory mice. And while those can be very informative, there's major differences between mice and humans, obviously. So we've also replicated our experiments using tissue from organ donors, from human patients who have passed and donated their organs to science. Typically, if eyeballs are in good condition, then their corneas are used for corneal transplants. in other patients that could benefit from that. But in some eyes, the cornea is not healthy enough to be used for corneal transplants. And so then the eyes go to science. And in our experiments, we were able to take pieces of human retinal tissue, transplant human stem cell derived retinoganglion cells onto that retina, and then compare what happens if you leave that membrane on the surface of the retina, that barrier, intact, versus if you digest it with an enzyme. And just like in the mice, we saw much, much better engraftment of these RGCs when that membrane was disrupted, telling us that as we are thinking about developing surgeries for human patients to actually transplant these cells, removing that membrane or that fence is going to be an important part of that. And indeed, that's what we're doing right now. So this is a movie of a surgery being conducted in a human patient by a colleague of mine, the director of the retina division here at Wilmer, Dr. Jim Handa. So this is a patient with an epiretinal membrane. This is a membrane that's growing on the surface of the retina where it shouldn't be, and it's causing blurred vision. And to treat this, he has stained the inner limiting membrane, that fence that we're so worried about, with a green dye. And then under the microscope, he's using a tiny little instrument that's just a little bit bigger than a human hair to peel that membrane right off the surface of the retina. So this is a surgery that's already being done in patients. We know that it's safe. We know it's effective. And so we are working on translating a version of this surgery in a model of glaucoma and ischemic optic neuropathy that we can use to start planning our methods for doing retinoganglion cell transplants in human patients. We've done some such studies so far. So this is a retina that comes from a larger animal model, one that has an eyeball that is much more similar to a human than a mouse. And we have done this surgery where we remove the vitreous gel. Dr. Handa removed the membrane from the surface of the retina, and then we transplanted red human retinoganglion cells onto this retina and then left it for 2 months. And after 2 months, we saw excellent survival of these transplanted retinoganglion cells. And remarkably, just like in the mice, these retinoganglion cells sent axons directly towards the optic nerve head so that these axons could go out the back of the screen here through the optic nerve and towards the brain. And interestingly, we transplanted cells in areas where Dr. Handa did not peel the ILM, that membrane on the surface of the retina, and in areas where he did. And the results were remarkably better in the areas of the membrane peel. So we're now very confident that not only is that going to be an important part of the surgery as we translate this to human patients, but we are confident that we have a safe and effective way of actually doing that membrane removal. When we look at the retinoganglion cells at higher magnification in this large animal model, we can see that the cells extend dendrites, and those dendrites dive down into the inner plexiform layer, that special layer of the retina where those input synapses from the other cells of the retina need to occur. So we're very excited. We have made a tremendous amount of progress over the last 4 years. And in some ways, we've managed to overcome half of the major challenge that exists in restoring vision for patients with glaucoma. But we have a big challenge ahead. We still need to figure out how to get those retinoganglion cell axons to go all the way through the optic nerve and into the correct locations in the brain. And once they arrive there, they need to form communication stations or synapses with the nerve cells in the brain. In essence, we need to achieve connectivity between the cells that we put in the eye and the brain. And not just for a handful or dozens of neurons, but probably tens of thousands of neurons. The other thing that's really critical to understand is that the connectivity needs to be organized. And so I want to go back to or end with one more kind of old school analogy for everyone here. In some ways, you can think of the optic nerve and its inputs into the brain as the kind of telephone switchboard that used to exist in the basement of large office buildings, where you'd connect people to each office phone line had a specific plug in this board, and if you plugged the phone cable into the wrong one, the person on the other end wouldn't be able to talk to the person they intended. In order for us to restore vision in a way that is meaningful, that actually produces a picture of what a patient is looking at, we need to be connecting each of these retinoganglion cell fibers to the correct socket in the switchboard inside the brain. So the final analogy here goes back to the TV that we were talking about. Where we are right now is we have patients with optic neuropathy that are completely blind. They have no ability to even perceive light, to tell if the lights are on or off in a dark room. That's like having a TV where there's absolutely no picture. If we're able to restore retinoganglion cells, but we don't pay attention to or have the ability to control the connections and the patterns of connections that they make in the retina and the brain, we might start to get light coming out of the TV, but it could end up being a scrambled mess, like white noise static on the TV. So how do we envision retinoganglion cell transplantation to proceed in the future? Well, this is going to occur in sequential iterative steps. And the first step is probably only going to be restoring the ability to see light. We might be able to turn on the TV, but it may not have a picture that's necessarily interpretable. Now, that in and of itself, I think would be a scientific achievement for sure. And I don't want to undersell the effect that this could have on the quality of life for patients. Patients that are completely blind from optic neuropathy or any other eye disease can't detect light, and so they have no circadian rhythm. And some of them suffer from a condition called non-24, where they actually are unable to regulate when they're awake and when they're tired, and they end up sleeping all day and they can't fall asleep at night. And this creates problems for them in trying to interact with the world around them. So even being able to restore the ability to perceive light could be a major advance and very important for patients. But our ultimate goals certainly don't end there. I think as we're able to continuously refine the connections that we form between the eye and the brain, we hope to be able to have patients gain some spatial contrast, to see that this part of the picture is a little brighter and this part's a little bit darker. And as that gets better, they might start to see that there's something dark in the middle of the screen and other parts of the screen are lighter and different. And this might start to slowly adapt into general shapes. You might be able to make out that there's a person sitting here in front of some kind of an object to his right. And eventually, as we continue to refine this, we hope to be able to one day provide vision that is at the level that really helps patients function, doing things like recognizing faces, reading letters and texts. That is our ultimate goal. We're shooting for the stars. But I think it's important to, at the same time, be realistic and understand that this, while it's our ultimate goal, will take considerable time and ongoing efforts. I want to end with just one other note. It's something that's important to me as a scientist. I am a strong advocate of collaborative and open science. A problem like optic nerve regeneration is not one that my lab alone can solve in its entirety. This is something that requires expertise from lots of different fields: electrical engineers, biomedical engineers, neuroscientists, clinicians. And so I, with help from several colleagues, including some at Wilmer, several years ago, started this consortium called RESTORE, which stands for retinoganglion cell repopulation, stem cell transplantation, and optic nerve regeneration. And this has grown into a group of scientists numbering more than 300 all over the world that are all interested in working together, sharing their discoveries, and collaborating to make vision restoration for optic neuropathies actually come to fruition. And so for the people on the call, I want to reiterate that there are a lot of brilliant minds all over the world working hard on this problem to eventually be able to improve the vision for patients and for the lives of their families and friends and caregivers. So that's all I have for the presentation. I do want to thank several people and groups here. Listed on the left are all the people, the scientists and the students in my laboratory who have done most of the experiments I just talked about. We collaborate extensively with Don Zak and Harry Quigley here at the Wilmer Eye Institute, as well as some other scientists here. I want to thank really the Zenkel family and Lois Zenkel for their generosity and also their foresight in making this work possible. Because this is something that I think we all understand can't happen overnight. But when it is successful, it will change the lives of millions of people all over the world. I also want to thank Alan and Shelley Holt for sponsoring my Rising Professorship, which has been also as transformative. And I want to thank, I know there are many people on this call, my patients, patients of others, friends and family of the Zenkels, who have also supported our work. And I want to sincerely thank everyone for that. I can truly say that where we are today is easily 4 to 5 years ahead of where we would be if we hadn't had all of this support. And so by so generously supporting this work, you and everyone else are helping the patients have to suffer with blindness for a shorter period of time than they otherwise would. So thank you very much. I will stop sharing my screen there. And we do have time for questions for anyone that would like to ask them.
Can you type? I'm going to tell you what you typed. Are there?
You can also ask it. Here's your.
I don't want to ask the question. I want to type.
Yeah, if you type it into the chat, I'll read it off. There are some in the chat right now, so I can get us started with that. There's a question: how important are amacrine and horizontal cells to the regeneration process? That's a pretty informed question. So amacrine and horizontal cells, I didn't even mention, but those are yet another subtype of neuron that lives within the retina. And they're important for fine-tuning the Morse code that comes out of the retinoganglion cells. Ultimately, horizontal cells live next to the rods and cones, and they're not particularly involved in the Morse code generation from retinoganglion cells, but amacrine cells are. And they're also, I skipped over it for simplicity, but they, along with bipolar cells, are the two types of retinal nerve cells that have to provide information to the retinal ganglion cells, the RGCs. And so they are the two types of cells that we're looking at when we are studying and creating assays of the input synapses or communication station of RGCs in the retina. Somebody asked what tomato stands for. So if you were listening closely, the human RGCs that we create from stem cells are tagged with a red fluorescent protein so that we can track them in microscopy and know which cells were the ones that we transplanted and which cells belonged to the mouse and were there since the beginning of the mouse's life. And the specific name for the red protein that we use is called TD tomato. Tomato doesn't stand for anything. I think it's just a cute name by the scientists that invented the gene because tomatoes are red and the protein is red. Is there any need to restore or repair the membrane after implanting the new cells? That's a fantastic question. There probably is. It's interesting. If all you were going to do is peel the membrane off the surface of the retina, you can leave it. Patients don't need a membrane on the surface of their retina. And we know that because there's been thousands and thousands of patients that have had surgical peeling of the membrane on their retina for other types of diseases that we didn't talk about today that are not optic neuropathies. And those patients not only do fine, they actually see better after that surgery than they did before. So we know the membrane is not critical to the function of a retina that is otherwise capable of seeing. However, the membrane is very important for the development of retinal ganglion cells and an optic nerve, and for telling the new retinal ganglion cells that have been transplanted into the eye where to make their connections. Which direction do they have to go to find the photoreceptors, and which direction do they have to go to get to the optic nerve? So, I will tell you that part of the project that we're working on that's funded by the Zenkel family involves developing a biomaterial scaffold in collaboration with biomedical engineers that allows us to transplant these cells into the eye. And that scaffold actually contains an artificial membrane so that we can peel the membrane, put the retinal ganglion cells down on the retina, and then have a new artificial membrane on top of it that will provide the developmental signals necessary for these new transplanted neurons to grow correctly. What role do you see AI playing in advancing a cure or quantum computers? That's a very good question. I think I see AI becoming very important when we begin to fine-tune the connections that are forming between the eye and the brain. When we go back to that telephone switchboard analogy, there are going to be hundreds of thousands of RGC axons that we're going to be getting into the brain. And then there are going to be hundreds of thousands of neurons in the brain that need to connect to those RGC axons. And they're going to need to do it in a very precise pattern that allows the picture that's formed in someone's brain to make sense. And I think at this point, we do not fully understand what the normal pattern of connectivity is. And it's hard to understand that on a global level because there's so many connections. Another news item that's been making the rounds in the last week has been the publication of the connectome of the fruit fly brain. So we've just now reached the stage where scientists have been able to map all of the connections that are present in the brain of a fruit fly. And just the tiny part of the brain that optic nerve axons connect to in a human brain is probably as complex or more complex than an entire fruit fly brain. But AI has been really important in helping us get to that stage because you need supercomputers essentially to be able to understand the billions and billions of connections that are made between all of these types of neurons. Do dead RGCs need to be removed to make room for the newly implanted RGCs? Also a very good question. I think that's probably true. but that actually is part of the optic neuropathy disease process. As retinal ganglion cells die as a result of the disease, there are other types of cells within the retina and the optic nerve and the brain that are called microglia and astrocytes. And together, those cells are sort of like the garbage trucks of the central nervous system. They wander through the brain and the optic nerve and the retina. And whenever they encounter debris of dying cells, they engulf it and degrade it. So the RGCs, we don't need to do anything to remove them because the body takes care of that to some extent naturally. I think, let's see. What can you tell us about what happens to the axons after they enter the optic nerve? Have you seen them growing through the optic nerve? Yeah, that's the question that when Dan Zenkel was visiting a few months ago, we were talking about. And we're almost at the point now where we have a really cool new scientific technique where we can answer this question in high detail. It used to be that in order to tell if there were axons or fibers within the optic nerve, you'd have to cut very thin sections of the optic nerve and put it on microscope slides. And one optic nerve might end up being represented as 100 different cross sections of the tissue. And then you'd have to look at all of those 100 cross sections and try to put it back together, the three-dimensional configuration of all these tissues. And it was tricky because if a fiber is going up and down and left and right within the optic nerve, and you were to cut a single cross section of that optic nerve, you wouldn't see a continuous line. You'd see little dots and little breaks and things because the fiber would be going in and out of the plane of that cross section. What we're doing now is working on a technique to turn optic nerves essentially transparent like glass. And so then we don't need to cut them into little pieces or sections. We can look at the entire optic nerve as a three-dimensional structure and image it using laser microscopy. So we had been working on that up until this point. We've now got it working. And so in just the next few weeks, we should be getting our first taste of what those retinal ganglion cell axons look like in the optic nerve. This will also tell us how fast they can grow through the optic nerve. You know, if somebody has a nerve injury in their arm, for instance, they might end up with numbness and weakness in their fingers. The peripheral nervous system, the nerves in your arms and your legs, regenerate naturally. And that's different than what happens in the nerves in your brain and spinal cord and optic nerve. But in somebody that's had a nerve injury in their arm, they'll have numbness and tingling for the amount of time that it takes for those nerve axons to regrow all the way to the tips of their fingers. And that's not a very fast process. That can take weeks or months in humans. So for retinal ganglion cells to regrow axons from the eyeball to the brain, it's not quite as long of a distance as going all the way down the arm, but it's several centimeters. And so that may mean that if we transplant retinal ganglion cells into the eyes of a patient, they may not be able to see anything for maybe 4 or 6 months after the surgery because it may take that long for the axons to grow all the way to the correct location in the brain. All right. I think I've gone through all the questions in the chat. Are there any other questions that anyone would like me to address? All right. Well, I did provide my email address. If anyone wants to contact me, I'm always happy to discuss by email or on the phone. Otherwise, I thank you very much again for spending the afternoon with us. I hope it was informative and hopefully exciting for the future. And
Can I comment briefly?
oh, please, yeah, Dr.
Yeah.
Quigley.
Hi, Harry Quigley. think that Tom underplayed the fact that he got 300 international scientists to begin working together and sharing information in a way that is not the usual academic mode. The usual academic mode is you carefully guard what you know and publish it first before somebody else. These restore meetings that Tom has gotten together have gotten scientists all to say, look, we have this amazing problem and we've got to solve it for people. And if we all sit there and hide what we know from each other, everybody's going to take much, much longer to get to a solution. And the fact that he's been able to do that and get all of these really smart men and women working together and holding meetings together is also thanks to the people who are on this call, because the cost of doing that kind of thing, while it isn't huge, is needs to be funded. So thank you all very much.
Indeed. Thank you, Dr. Quigley. All right, Kathy, I'll turn it back to you to sign us off here.
Well, again, thank you all for joining us. Hopefully it was as educational for you as it continues to be for me. We wish you all well. And again, if you have anything that you might need from us, please don't hesitate to reach out. In the meantime, take care. Thanks again for your time and, of course, for your support.
Thanks, Tom.
Thank you.